decades ago. It's a great pleasure to have him here, and the title today is Modeling Gene Expression and Regulation in Response to Light-Dark Signaling. So, Nick, thank you very much.
Thank you. I really appreciate that introduction, but you might be setting me up for failure a little bit here, because one of the reasons I wanted to use this project for my seminar today was to introduce you to my current research, because it didn't go perfectly. Lou's right, we really wanted to get at that question of what is the underlying regulatory basis for how genes respond to light-dark signaling, but we kind of, we hit a little bit of a hitch about halfway through. So I won't spoil any more there, but really this project is about how my research started, how I got onto the question I'm looking at today, and I'll touch on my future directions and the projects I'll be working at here at Nimbus just a little bit at the end. So, the overall focus of my research from MSU through now has been understanding the function of differential expression and the systems responsible for regulating it. So the first thing to explain here is, what do I mean by differential expression? And the best way to do that is by comparison. So I'm hoping that most, if not all of you, are familiar with Mendel's experiments, sort of the basis of genetics as we know it today. In this case, the difference between these two P's, this round yellow P at the top and this crinkly green one at the bottom, is that although they have the same genes, this R locus and this Y locus, they have two different types of alleles that are responsible for the difference. Comparably, you can look at two different cell types in the human body, these epithelial cells, which are adhering to one another and stationary, versus this mesenchymal cell kind of floating at the top, not adhering, more mobile. And the difference here is not different genes or different alleles, it's gene expression. We have identical genomes which are doing different things. So what is that difference? Well, in the epithelial layer, you're expressing more of this protein called E-cadherin, which is in part responsible for that adhesion, and you have none of this vimentin. While in the mesenchymal cell, E-cadherin goes down, vimentin goes up. There's more than just these two genes responsible for it, but it's this expression difference which we use as the marker between these two cell types. And if you need any further proof, you can actually interconvert between these two cell types, and the expression of E-cadherin will go up if you go to epithelial cell and go down if you go to a mesenchymal cell. It's a dynamic change that's responsible for this difference in, maybe not phenotype, but difference in character. So, why is this going on? Well, if an organism can express different characters by having different alleles, why do we need this more dynamic differential expression? Well, organisms face many different types of signaling. Broadly, you can think of them as being internal or external, so you can have things like the day-night cycle or the cell cycle, and they can also be periodic, like the two cycles I just talked about, or very acute, such as interaction with a predator or exposure internally to some sort of hormone or signaling molecule. Now, for us and for other creatures which are more mobile, which can alter their environment, you can think, well, if you get an environmental signal, you move or you change your behavior. But for some organisms, particularly plants, yeast, algae, they are limited in the way they can change their environment. They're not perfectly immobile, they're not perfectly stuck, but they have less options. So many of these reactions to stimuli tend to occur at the genetic level. So we've defined differential expression, we kind of have an idea of why it might be occurring, so how do we approach and study this? Well, we can approach differential expression at three different levels. First, and most simply, we can just measure the expression of genes and look at their patterns. So we can see here, some might be going up, some might be going down, some might remain stationary, and some may have more complicated behaviors. Once we've established what these expression patterns are, we can then start looking at regulation. What genes, we would call them transcription factors here, the lettered ones, are regulating the genes which are producing these different patterns of expression? And more importantly, what differences in regulations might be responsible for differences in patterns? So example, both our blue and green genes are regulated by C, but they have different expression patterns. So Maybe it's the effect of D and E on top of C, which is why blue is different from green. And finally, although I won't get into this much today, there's the evolutionary question. For our regulators in particular, we know they tend to be duplicated a lot. So how did the family of regulators we have arise? What was kept? What was not kept? What tends to be the properties of these regulators, which then go on to create this wide spectrum of expression? So this is ideally how we would like to approach the problem. And as I said today, I'll mostly be focusing on expression and regulation. But if it was this simple, you wouldn't need science, you'd just be able to look at stuff. So what are the challenges we're facing? The particular challenge is expression comes in different patterns. And it might be easy to look at this sort of idealized figure and say, oh, it's obvious what's going on. But if you only measure expression at one time point, now it's not so clear. Is blue being highly expressed, or is it stationary and just higher relative to something else? Is red really going down, or is it just at a particular part of its cycle, which would change if you measured it anywhere else? This is further complicated by the fact that there's noise in our measurement. So these patterns aren't going to be obvious even if we have the convenience of looking at them. Now, and you might say, okay, noise is a part of life. You're always going to get some things wrong. You'll misclassify something as high, low, or indifferent a certain percentage of the time. Well, even if that was true, you still have this cycling pattern. And that's not a quantitative issue, that's something which is qualitatively different. So is this something we really need to be concerned about when we're looking at expression? The unfortunate answer, or fortunate, depending on how your career is based, is yes. So on the right here, we have a diagram of the circadian clock, particularly the one from Arabidopsis. This is an interlocking system of regulatory genes. You can kind of think of it like one of those cast metal puzzles where you're trying to pull the rings apart. The key feature of it is this can cycle on its own. Just left to its own devices, some genes will go up and then go down, antagonistic to their, the ones that go in the opposite direction. However, it also takes input from external signals like the day-night cycle, like hot and cold. The sum total of this is in Arabidopsis, the model plant species. About 90% of the genome cycles in response to this clock under some set of conditions. Now, in some cases, the cycling processes are pretty obvious. Unsurprisingly, photosynthetic processes cycle. Observations of this go all the way back to Alexander the Great, where one of his generals on campaign sat under this tamarind tree and noticed how the leaves move with the sun. So there's some things we expect to cycle. However, consider this green algae. It's also photosynthetic. We also would expect that to cycle, but it's a single-celled organism. We don't have a lot of structures we can look at and interpret what other things may be cyclic. We have to approach this on a genetic level. So that's the setup for the problem. And I'd just like to quickly define the objectives we had going into this study. First of all, we wanted to be able to identify cyclically expressed genes from expression data. This may seem pretty obvious, but I want to highlight we want to do it from expression data. We do not want any pre-existing information. We want to be able to look at a pattern and say, this is or is not cycling. Because again, you may have a tree where it's obvious, or you may have an algae where you can either observe it under the microscope or you can sequence it. You don't have as many options. Now, once we have identified something as cycling, we don't want to rest on our laurels. We don't want to say, well, it's numerically significant, we're good. We want to be able to demonstrate that there's some biological significance to these patterns we've identified. Finally, once we've identified patterns, once we've proved they have some connection to some biological function, we want to identify what's regulating that expression and ideally construct a predictive model so that we can say, no, it's not just this regulator attaches to this genes. It's if you have this set of regulators, I can distinguish cycling from non-cycling, or going forward, cycling at one time point from another. So that's our setup going into this study. So first of all, how do we approach this? Well, we decided to use that green algae I showed you before called Chlamydomonas reinhardti. It's, again, another model organism as our organismal system because it's a photosynthetic algae, it's well studied, it's a model. But one thing that's really was never done to it before this study was a genome-wide approach to understanding cycling expression. There'd been some small case studies. They'd showed, okay, it can happen in something like photosynthesis, which we expect, but really genome-wide, what's going on in this little single-celled organism. Additionally, we know that there should be something cycling. Obviously, photosynthesis and the Calvin cycle, but additionally, there's some physiological evidence that genome replication and, in fact, the movement of this algae is biased to happen either during the day or the night. So, to explore these types of cycling expression patterns we expect to see, we measured expression under light-dark conditions. So we have basically a data set sampled every 3 hours from 0 to 48, which cycles between 12 hours of light and 12 hours of dark. And ideally, what we would like to see is expression patterns which are repeatable over that time course and ideally rather symmetric. So they peak at about the same point at the same place in either day. Now, obviously, if I'm showing you this as my ideal, I'm making an assumption about what cycling looks like. I'm basically saying it's approximately a cosine wave. And this is not necessarily true. You can have other patterns of cycling, but this is a good place to start because it allows us to define properties of the type of behavior we expect. So, treating, for the moment, cycling expression as a cosine wave, we can define, first of all, that our patterns will have a phase. This is the peak. This is basically the delay after time 0, which we see our first peak. And this is going to be probably the most important property we talk about because this is what's going to allow us to differentiate between different cycling patterns, whether they occur early or late during our time course. Additionally, we can define the period, which is the time between two peaks, or you can think of this as the time which the cycle repeats itself. This is important from the modeling aspect because if this is a day-night cycle, we expect this to be about 24 hours. And we're going to use that a lot when we're defining our models. Finally, and we're not going to talk about this much, but it has its place, the amplitude. And that's going to be the height of this peak relative to where the average expression level is. We don't do a lot with this in terms of exploring the organism, but this turns out to be very important for evaluating your cycles. Because mostly what we're going to be doing is comparing the amplitude of a model to the amplitude we get by just randomizing our data. All right, so we started off looking to see what exists in terms of modeling cycling expression in organisms. And the first thing we stumbled upon was this program called, and I think it's COSOPT. I think it's short for cosine optimization. This is something that the authors kind of left you to infer, so I'm not really sure. But with a name like cosine optimization, it makes sense that really what this is doing is it's fitting a cosine wave to our data. We're defining our expression x as a cosine wave with a particular phase and period and amplitude plus some mean expression value. And you can think of this graphically as we have some sort of expression vector we're observing, and we're just trying to find the best fit cosine wave for it. The way the program does it is you give it a range of period and phases you think are relevant. It does a grid search for all of those combinations and optimizes the amplitude and mean for every basically node on that grid using least squares. You find the best fitting model, you compare the amplitude of that best fit to just taking all the expression data you give it, shuffling it up, seeing what that produces. And at a false discovery rate of 5%, we correctly identified 5 of these 15 gold standard genes, which we have physiological data we know cycle. So we don't know how many cycling genes there are in this organism, but 7 out of 15 isn't a good start for comparison to our gold standard. So we looked, we tried some other programs. They're basically all doing model fitting, and eventually it came to the point of, okay, maybe the problem isn't this program, maybe the problem is the approach. Because here we're assuming that everything that cycles is approximately a cosine wave, and it looks like that. What if instead we use something like the discrete Fourier transform? And what this does is we're not actually modeling expression here, we're converting it. We're taking all of our expression values, our x for n, so our x over time, and converting it to a new value, which is x over k. And k here you can think of as our period. So for any cycle, you can imagine it's made up of a bunch of different components. Some don't cycle, they have a period of 0, they're constant. Some occur once a cycle, or twice a cycle, or 3 times a cycle. So we're basically saying, how important is cycling at every period to this expression vector we gave you? Graphically, what we're doing is we're taking this expression vector and converting it to this. So here we have the amplitude over the different possible periods in our expression vector. So 0 is our baseline, 1 is once per period, basically once per vector, 2 is twice per vector, and so on and so forth. And you can see that it's symmetric around 8, and that's because basically the periods beyond 8 are harmonic with the ones before. So 2 and 14 match up, 3 and 13 match up, so on and so forth. The important thing to notice here is, remember I gave you a expression vector that's supposed to cycle daily over 2 days, so as you would expect, the amplitude for the period of the whole of t over 2, of the whole cycle over 2, is large. And this is the approach we used to identify here. For those of you a little more familiar with Welch's method, or with DFT, we applied Welch's method, we normalized the output, and we got a score. Again, same thing as with COSOPT, we randomized our data, compared it to the population, got an FDR of about 5%, and this time we got 8 out of 15, so not much improvement. So we scratched our head for a bit, thought about doing a new method, but eventually decided, okay, before we throw all this out, let's look at our scores. How do they compare with one another? And what we found was, even though they're highly correlated, got an R squared of 7.6, there's a lot less variation between low scoring genes than high scoring genes. Basically, a low score, so a poor Fourier transform score and a large p-value, tend to be very highly correlated, whereas you can have something with a small p-value and a much larger range of discrete Fourier transform score. So the way we decided to exploit this is, let's make our method inclusionary. Let's say we'll call you a cyclic gene if you pass either one FDR threshold or another. And we had to adjust this a little bit, basically, to make sure that the total FDR was 5%. But the long and the short of it is, if you use these methods in a combined way, we can get more of those gold standard genes. And basically, the final outcome is after we tweaked the thresholds a little bit, we got 11 out of 15. We could have got better, but basically, relaxing the thresholds would have got us up to 13 out of 15 gold standard genes, but it would have really only had a minor change on the population that we were calling cyclic, while exposing us to a lot more potential error. So if you want to ask questions about it, I'll be happy to field those later. But for now, the key point here is, by combining these two methods, we were able to basically get a lot more of our gold standard set while preserving the same false discovery rate. So what did we find? Well, overall, about 8,000 genes and Chlamydomonas cycle under light-dark conditions. This is around half the genome. I don't like to usually use specific percentages here because, again, there's some variance depending on what threshold you use. But as I said before, right now we're just relying on this being numerically significant. The cycling genes actually look like things that would cycle in response to this circadian clock in an organism. So we looked at some of the basic properties of it and found agreeable things. First of all, the average period for our identified cycling genes is about 24 hours with a standard deviation of 1 hour or more. This is a biological system. We don't expect things to be perfectly 24 hours. The important thing here, though, is we have this nice bell curve. We don't have a uniform distribution across all periods. There's a bias towards the period we would expect. Similarly, when we look at the phase of expression, remember now this is when the peak expression occurs, we find that there's basically 2 points of bias. One at 0 hours, so that's basically at simulated dawn, And the other, it's a little harder to see, but it's there, at basically transition from light to dark, at the simulated dusk. And what's important about this is this demonstrates what you would expect to see from the circadian clock, which is anticipatory. It's not that light occurs and then your light genes turn on and build up. It's things that need to be on during day or night anticipate that change and are already increasing or peaking when the change occurs. So our population, at least from these, in this very basic way, behaves in the way we would expect. Furthermore, we wanted to make sure that the models we have for these cycling genes, at least in some way, correspond to the expression data. So our model can predict, a wide variety of periods and phases, but realistically, we only sampled expression every 3 hours. So we took our models, sampled them in the same way, grouped them by where the peak was according to these time points, and lined it up with the actual expression data. And now you can see our expression data is much more noisy. It is not a perfect representation of the models, but the models in general get it right. So things which we put on our 0 phase cluster tend to peak around or maybe just after 0. It's hard to tell if we're really predicting the phase off or if it's just the fact that our data is noisy and it's not completely symmetric. But we use this as a justification for moving forward using these phase clusters to basically divide our cycling genes up and explore them in more detail. And one of the first things we did with these clusters is see if there's any functional significance to them. So we did a Go analysis, and we found 236 Go terms that were enriched in at least one of these clusters. But most importantly, of these 236 terms, over 90% were specific to a particular phase. So we find genes with this one particular function in the 0-hour cluster, the 3-hour cluster, the 6-hour cluster, but not in other phases. And if you allow some variance, like basically one phase or a neighboring phase, this comes up to 99%. So there's a very tight correlation between annotated function and the phase at which these cycling genes are being predicted. Or sorry, yeah, the phase would, yeah. Furthermore, if you take these Go terms, which can kind of at times be a little bit overly specific, and group them into sort of broader functional groups, we see the same pattern. So Go terms which are related to ribosomes or translation are all enriched at 3 hours. One's relating to photosynthesis and light response at 3 to 6, so that's basically morning and noon, as you would expect. Mitochondria at the same time as photosynthesis. And then what's I thought was the most interesting part, the cell cycle, basically all those genes are enriched for peaking at midnight, and the microtubules and flagella right before dawn. And the interesting thing about this is it is believed that Chlamydomonas preferentially divides at night to avoid possible damage from UV rays. And what this division involves is reorganizing all your microtubules, getting rid of all your motor proteins so that you can use those to separate the chromosomes and then divide evenly. Afterwards, you have to remake all your motor proteins. But there's a slight delay between when the timing of replication occurs, because you don't need that motor again until daytime. So basically there's this nice sequence of, okay, you have to break down your motor to divide, but you don't need it again till day, so let's wait to reassemble it until right before dawn, until right before you need it. It's that key anticipatory behavior that you see from the circadian clock. All right, so here we from the Go term perspective, found enrichment of these functions with phase, but is that actually reflected in expression? So here we took all of the genes that fell into each one of these function groups, no matter whether we predicted them to cycle or not, and looked at their average expression. And although there's a lot of variance in that expression, the gray bar here represents ±1 standard deviation. In general, the predictive phase is where these groups peak. So we not only see functional enrichment, the phase is actually a pretty good predictor of the general behavior of these gene functions. And now one final piece showing the significance of these identified patterns before we get into their regulation is we looked at the evolution of cycling by building a phylogeny of genes in Chlamydomonas, picking out basically the leaf pairs where we could find them, and looking at whether or not both genes cycled, didn't cycle, or were mixed. And our first observation here was that we argue that this shows that in general, cycling genes tend to be more conserved, because if you look at the frequency of cycling duplicate pairs across the synonymous substitution rate, which is what we're using as a proxy for time, it's relatively stable, whereas your non-cycling genes start off much higher. They're proportionally more frequent in these younger duplicates. But over time, it seems to be that they tend to diverge, where one of them will pick up cycling behavior. And so if our assumption here was, if you treat this as sort of a snapshot of what may have occurred during time, the only way you can explain this difference is a bias towards the loss of non-cycling genes. This is something I would have loved to expand on, basically by doing ancestral inference, but we didn't have the opportunity. More importantly, though, whether or not I can convince you this really represents cycling as conserved, if we look at duplicate pairs where they're both cycling, the tendency, if our first duplicate has a phase of 0, is that the other duplicate will either also have the same phase, or that its deviation from 0 will be small, 3 hours in one direction, and not large, i.e. 12 hours, so the maximum possible deviation you could observe. So this feature, which we found previously was associated with function, also appears to be conserved between duplicate cycling genes. So given that, we concluded that these phase groups are in some way significant, so we decided to try to find regulators and explain the regulation of the phase of cycling expression. And to do this, we first did motif discovery within each phase group. And what this involves is taking the promoter sequence of all genes in the group, giving them to a set of programs which then find enriched motifs. So these are small sequences of bases which occur repeatedly in the promoter region. We then cluster these motifs because the same program could find the same motif or find similar motifs. We build clusters of those similar motifs, and then from them derive a consensus motif where each position is not a single base, but a probability of being a certain base. And that's represented here by the height. So in this consensus motif down at the bottom, what we're saying is that first position we don't really know, it could be anything, but the next position is likely G, followed by a T, followed either by a CG, followed by a C. So we can use these consensus motifs to represent different putative cis-regulatory binding sites in our different phase groups. And once we have these putative sites, we can start to build a model to explain the differences in regulation of the phase groups. And we used two different approaches to modeling them. One, naive classification, and all this means is if you don't have a motif, you don't cycle at the phase. If you have a motif that was enriched in that phase group, you cycle at that phase. And if you have motifs from many phase groups, we predict you cycle at multiple phases. Even though this is unreasonable, we still use this because it's a good baseline to see how much information are we getting just from motifs alone. And instead of trying to think of more complicated rule sets we could use in this classification, we also employed a machine learning method called SVM, or support vector machine. And the easiest way to think about this is, let's suppose we just had two motifs, and we're trying to explain the difference between the black and the white phase groups. What SVM is going to try to do is weight the presence or absence of these motifs to define a plane separating these two groups. Same thing is happening with our data, except instead of two dimensions, instead of two features, this machine learning method is going to try to build the plane across multiple dimensions. And in doing that, it's going to weight and identify different motifs that are more or less important to different phase groups. And there's a lot you can do once you get past this step. You can build, once you have basically a set of motifs you think are important, you can start exploring connections, you can start trying to connect them to specific proteins. But I'm not going to go into detail here because this is sort of where the stopping point for our study was. Because although we found enriched motifs in every phase group, and in general these motifs tended to be pretty specific, although there was some overlap between 12 and 15 hours, so right at that day to night transition. When we went ahead to predict this, the results weren't great. So what we have here is the evaluation of our model for each cluster using something called AUCROC. And there's a big graphical explanation of what this score means. But the easiest way of thinking of it is the probability that that a gene with the desired phase has a higher score in the model than one without it. So if our model was perfect, all the genes with a specific phase would rise to the top, kind of like cream. The rest of them would sink, and any good gene that you found would have a higher score than the rest. Alternatively, if they're completely intermixed, the probability of one scoring being higher would be about 50%. That's the worst you could possibly do. So, as you can see, all of our models did better than that 0.5 score. And our naive had an average somewhere around 0.6 to 0.8. The SVM model, the one that actually weights different motifs, performs better in every case. But the problem here is this wasn't what we'd expected. Now, we're not Amazon. We don't want 0.9. We don't want perfect predictions. But when we'd applied this same strategy to predicting what I'll hesitantly say a simpler system, like just response to a specific stress, just response at one time point, we got much better scores. And we were able to leverage this higher confidence into doing more detailed exploration of the regulatory system. So, our immediate question is, what went wrong here? We tried a couple other different algorithms. It didn't seem to make much sense. Our initial thought was, well, maybe our phase groups are too broad. Maybe this decision that everything that's within the same 3 hours is the same is arbitrary, and it's not what's happening with the plan. So we said, okay, first of all, let's further cluster our phase groups so that we have things that are not only expressed at phase 0, but are actually correlated with one another beyond that. And we found, okay, 28 of these clusters we made could be predicted with an AUCRC above 0.7 within that range that we previously saw. And this represents about 20% of our genes total. And as you can see, if we look at these co-expression clusters, they tend to be pretty nice. They are very well correlated in their expression. The problem is, if we look at the 28 worst clusters that perform lower than any individual model, they also seem really well co-expressed. There's not a bias in these clusters towards whether or not, let me take a second. We can't explain why some clusters perform better or worse based on how well our clustering method put them together and based on how well correlated they are. So even though this method worked, there's not a biological reason that we can see why. We can't point to a reason. So something else we did is said, okay, maybe it's not closer expression, maybe it's something else. So let's try breaking down these phase clusters by different functions, because we know genes that have the same phase don't necessarily have the same function. And again, we could find a few clusters here, 12, it covers around 10% of the genome, that we could also predict in our desired range. But now, their co-expression isn't necessarily that great anymore. Particularly this nucleolus cluster, the average expression is about 0.5 across every time point because it's all over the place. Yet, the enriched motifs that we found that were phase specific still do a good job of predicting it. And that's basically, unfortunately, where we ended. Because we showed that our method could work, we could predict stuff. There's obviously some information in these motifs, but we can't point to a good biological reason about why it should work. We are missing something that's key to this modeling process. So, for this paper, the conclusions were We can definitely identify cycling genes. About half of the genes in Chlamydomonas, this key model algae, turned out to cycle in response to light dart signaling, and we could tie the phase of that cycling to function and show that to some extent this phase is evolutionarily conserved. However, the regulation of the cycling phase remains an open question. So this is where my first project ended, and for the next about 3 or 4 years, as I did other things, I was wrestling with this question, which eventually evolved into how do organisms regulate timed expression? Any expression where timing is key, because one of the first things I looked at after this project was, well, maybe we need a different organism. We looked at Arabidopsis, that model plant. We tried looking at circadian expression there, no improvement. We even tried going back to a system we knew and taking stress response and saying, okay, forget stress response in general, what responds early, what responds late, what responds in the middle. And even though this system worked for general stress response, as soon as we tried to make it timed, same results. Okay, so maybe it's not the organism, maybe we need better data. Maybe this motif inference works for something very general, something very broad, but when you get down to timing, let's change over to a protein binding microarray where we have actual evidence of interaction between our regulators and our gene. Same result, not much improvement. Well, okay, maybe we need a better system. This machine learning stuff, it's up and coming. There's new developments all the time. Let's do random forest. It's provably, well, some would say provably, some would say arguably better than SVM under a lot of conditions. Same results, no improvement. Eventually, we kind of threw everything together. We went to Saccharomyces cerevisiae, because it's well studied. It has protein binding microarrays and chip-chip. We used a better random forest algorithm. We even went so far as to say, okay, let's not just use individual regulators, let's start putting them together in little motifs like feed-forward loops and feedback loops. And we finally got a sense of the results. We finally got something publishable. And the reason I didn't present that as presented this is I think the important part was the journey to get there, to realize, okay, yeah, you can eke out some explanation of how expression is regulated using this approach, but really what got us over the hump in the end was modeling, was looking at the detail of how different things interact. So that's actually what led me to Nimbus. Is I finished that last project. We'd done modeling on a very small scale. We'd identified small motifs, but I wanted to start approaching this problem from, a large, comprehensive model about how expression timing is controlled. So towards that, I'm going to give you just a brief intro into the 2 projects I'm working on currently. One is with Tian Hong, who is my primary mentor, and he has this very nice model of that first system I presented you, of the transition between an epithelial and a mesenchymal cell. So we have these regulators, TGF-beta, snail ZEB1, our regulatory RNAs here, that form a system of interactions which can explain the change in cell type using these 2 marker genes I talked about before, E-cadherin and vimentin. And you can not only capture the regular transition behavior, but you can recreate different sort of aberrant behaviors like stopping halfway through a transition or having a less committed transition by knocking out the components of this model. And what I would like to do is say, okay, since we know this captures cell type behavior overall, can we extend or use this model as is to explain not just these marker genes, but the whole cassette of genes which are responsible for that difference between a fixed adhering epithelial cell and a mobile non-adhering mesenchymal cell. And that's actually what I'm working on right now. And although it would be very nice if just, all the epithelial genes are kind of like E-cadherin and all the mesenchymal genes are kind of like vimentin, one thing we ran across is that for our mesenchymal genes, there seems to be 2 categories. One of which, as soon as you introduce TGF-beta, its expression change is committed, and another one which is a little bit more facultative, which seems to be dependent on this ZEB1 activity. So what we're trying to do is we're basically still at that first step. We know that the differences in expression is important, but we're trying to define the groups. So what is the spectrum of M genes that we should be interested when we're trying to predict these changes? The other project I'm going to be working on is with both Tian and another researcher here at, I just forgot the name of this university. University of Tennessee at Knoxville, at UTK, Albrecht von Arnim. And this is kind of going back to my first project, because I'm again going to be looking at circadian and diel regulation, but of translation and not of transcription. So here we have a ribosomal protein, RPS6, which is its phosphorylation, its activity is under the control of both the circadian clock and directly through a light sensing pathway. And that's known, that's pretty well established. The thing we're interested in is this protein undergoes a phase change under certain conditions. It normally peaks during the day, but you change the conditions of temperature and heat and sugar enough, and its phase won't wander, it'll do a complete shift. It'll advance 12 hours forward into the night. So what we want to understand is, forget that basic model of the clock, what of all these possible interactions between the clock and light and sucrose and other pathways can explain this shift? And from there, what's regulating this shift and what could potentially be the reason that this shift is necessary? So, oh, yes. So, with that, I'm going to conclude my talk by just thanking the people who were involved in the research project I presented to you. This is the Shoe Lab up at MSU. We don't actually have 30 people. We have about 6 people with a large collection of dogs and family member and small children. I'd also like to thank the 2 people I'm working with here at Nimbus right now, Tian Hong and Albrecht von Arnim, as well as the NSF, MSU, and Nimbus for funding my research. And of course, all of you for being here today and listening to my talk. And with that, I'd like to take any questions. Use the mic. Okay. Cool. It should be on right now.
So, anybody want to start?
So I'm not 100% sure what part of the first work you presented would this be relevant, but I wonder if you've explored how much information you actually feasibly can expect to pull out of this kind of data, right? Given that then there's noise in the measurements and the fact that you have limited number of observations, like, is what you're seeing actually the best you could potentially do, getting 60, 70% of the cycling genes?
So that's a great question. So the question is, well, basically, the model performed what you would call poorly, but really what should you expect it to do? What AUC-ROC would you expect to get out of a model like this? I wish I could answer that in an analytical fashion. In truth, my representation of it as poor is based off of other work done by our lab. So in addition to stress response, we've also looked at things like, in addition to cycling, we've looked at stress response, we've looked at combined stress response, we've even looked at things like predicting regulations and genes involved in lethal pathways versus non-lethal pathways. And in those cases, the performance, you can always achieve a higher performance. 0.7 was the minimum, in some cases you got as high as 0.8. So there's this sense that even though this is a different system, if you approach regulation in general as a question, you can do better. You can maybe not get it perfect, but some of the key things were with these stress studies is you're able to get much better models. Then when you start digging into those models and looking at the importance of different regulatory sites, you can pull out these important sites and tie them back into, oh, this site is for this transcription factor. This transcription factor is maybe something we knew, or maybe something novel, or maybe it predicts an interaction at this promoter which we didn't know of before. With the type of modeling we had, it was so poor we didn't have any confidence in what it was telling us. We tried to pursue some of these regulatory sites and tried to connect them to different regulators, but just nothing came of it. So yeah, maybe that is the best you could do with the data we had, but it wasn't good enough.
Well, you had similar data and you were able to get substantially higher scores.
We had
Gene expression data, but asking a different question.
yeah, we had gene expression data. I think the key change though was we identified, we still use gene expression data to identify cycling genes, but instead of inferring regulatory sites, we had a combination of inferred sites, of sites predicted by PBMs, and I think the key thing was chip-chip data. Chip-chip on its own outperformed any other single data set, outperformed this study, and it was only slightly worse than using things from multiple data sets. And that's kind of a double-edged sword, because what that's saying is you can totally do this if you have the right data set, but it also tends to be the one where, well, you'd have to chip every transcription factor, which is feasible maybe for something like yeast, but humans, Arabidopsis, who knows?
Other questions?
So this might be my own ignorance, but do you think part of the difficulties in discovering those motifs and applying the machine learning algorithms that you used might lie in the fact that you can have these cyclic cycles over multiple timescales? You were essentially measuring things at a diurnal scale, but you could have seasonal scales. They may not be so relevant for a lab organism, but that would certainly complicate things if it was present.
That is a great question, and we know that is one of the problems we had. So we repeated this same analysis for another uncharacterized algae called Nanochoropsis, and basically because we had more money there, we got to dig into it a little bit more. Because in Nanochoropsis, we again had a gold standard set of genes, and there was a few that were missing. And were missing for reasons we couldn't explain easily. And what it turned out is, well, some of them were missing because they were actually cycling at a 12-hour period in just enough of a way that our program was having trouble understanding what it should do with that middle peak, because both of them, you kind of tell it what period to expect beforehand. The other thing that we found we were having trouble identifying was actually because we model things as being a cosine wave, which is nice and symmetric and very smooth. There was a set of genes in Nanochoropsis. That expression basically was flat and then spiked, and then flat again and spiked again. That's a problem we never really solved. So if you think there's multiple cycles, you run the model again. The only way we figured out how to find these spiked genes was basically doing differential expression point by point, and it having to be significantly up at one point and significantly down at every other point, you know, with a certain magnitude. It was very messy. So yeah. Our assumptions are limiting. Some of them are easier to overcome than others, though. If you like, period is easy, but if you have very atypical cycling patterns, that's much more difficult to deal with.
Okay, on that last case, so instead of a cosine type, you could, of course, just use a model that's flat and sudden.
Yes.
Flat and sudden. Did you try that?
So
that would pick out this peak, okay, to some extent,
Yeah.
if it was just after or just before a shift in light.
We had originally, we tried using another program called Haystack, which allowed user-defined models. And I think it's a fine program. I think the issue with it is not how it was made. It was the lack of a user manual. Maybe I'm being overly generous. No, but like I think I don't want to impinge the researchers or the data they came up with, because it was great, but they designed their models to fit in a 4-hour space framework, and I did what I could to adapt it to 3 hours, and it was off every time.
I see.
There's another program called if anyone wants to do this, called JTK cycle, which takes a very different approach to, it's basically a parameter-free version of a cycling model. It doesn't care how it looks, it just says, well, is the order such that it would cycle at about this period? It's got rave reviews. I've never been able to make it work for what I'm particularly trying to do, but I put it through some tough. I only ever tested it on the toughest cases I had. I think in general that'd probably be a better way to go right now than cosine fitting.
I see. And then one more question. The very last case, you said that you can get it to actually shift. So shift from light dark to the reverse. In the Clamamonas example, would you expect that there are some things you could turn on, turn off to cause essentially it's a phase shift 12 hours?
That's a good question. 12 hours, I'm not so sure. That's why I'm really interested in this project we're doing. Because it's easy to say, well, I could definitely turn off the cycling of photosynthetic genes by just not having light. I can very easily turn off the cell replication part by either making it always light, or in some cases you can just feed clammy so much it'll wait and it'll wait, and instead of dividing once it'll just divide like 8 or 16 times. But in all those cases you're just ablating cycling until you start it again. It'll eventually resume, and it will resume at a roughly the same phase. Getting that 12-hour phase transition is not something I cannot think of a good example of how that would happen.
Okay. I was just thinking that there might be some, mutation you could enforce through radiation or something that would affect certain components of the regulation, and that would cause a flip. In other words, use that to tease apart what's going on. And so.
Certainly. Like you could change enough about the circadian clock to flip it. I think one reason we might have not, one reason that would be a difficult route to go is they're so interlinked. So you might have to make sort of like, okay, you can change the time of this one gene, but the other gene is still going to be affected in the same way and entrained the same way.
So they're linked.
Yeah.
Okay. Any other questions? If not, let's thank Nick again.
Thank you.